to this webinar. I am Frank Zerbib. I am the current president of the European Society of Gastroenterology, ESNM. ESNM is the organizer of this series of webinars on the topic of Microbiota and Clostridium difficile infection. Today, this webinar is co-organized by our Canadian colleagues, the Canadian Association of Gastroenterology and the Canadian Neurogastroenterology Network, because it is the current name, but it will probably change in the next few weeks or months. So it's my pleasure to introduce you my co-chair, Dr. Louis, who is from Toronto, Canada. And we will have tonight, I say tonight because in France it's 1 a.m. So sorry, but we will have two talks. One will be a live talk and the second one will be a pre-recorded talk. So we will start with the first talk by Dr. William Turpin from Toronto, Canada. His talk is entitled Microbiome in Health and Disease. So William, you have about 20-25 minutes. You have the room.
Thank you for the introduction. Yeah, thank you very much for the introduction. It's my pleasure to be here today. Thank you. It's a great topic. It's going to be challenging to cover the human microbiome in health and disease. And so I'm going to start this talk. All right, so the human microbiome is quite complex in nature. And this is a slide that is a couple of years ago that just to show that there is still novel research going on. And at the time, it was thought that the human microbiome was representing the human genome in general. And at the time, it was thinking that there was a 1-to-10 ratio. Nowadays, it's more of a 1-to-N ratio between the human cells and the bacteria that we have in our guts. But what is still true today is when you look at the human genomes, we have about 23,000 genes, while the microbiome have 2 to 3 billion genes. So it really highlights that the microbiome is involved in many events that help humans to stay healthy. And it's involved in digestion, immune maturation, drug metabolism, and immune maturation. And so what is quite interesting is when we look at the microbes of a human being, you can identify a few taxa that are common in more than 95% of the population. So this is just a distribution that there is 2 major phylum in the human microbiome. Those are Firmicutes and Bacteroidetes. And those are specific genera that are found in most of the population. So you can see things like Bacteroidetes or Faecalibacterium that most individuals would carry at different degree of abundance. And there are also other phylums present in the human microbiome. This includes Proteobacteria, Actinobacteria, and so on. And in this study, that was comprised of about 20,000 individuals. When you look at microbiome diversity, which is a rough number idea of the which type of bacteria, the number of species that you can find, you can see as the sample size continue to increase with more than 20,000 individuals, we still don't reach the full plateau, meaning that there's still an increase in alpha diversity as the number of individuals continue to grow. With more than 20,000, we still don't fully understand or have characterized what is the microbiome diversity. And this is quite an issue when you consider that the microbiome is almost considered as a forgotten organ, and it is sensitive to different elements, including the environment, our diet with fibers, type of lipids that one may ingest. Those are metabolized by the microbes, and the microbes produces molecule that can interact directly with the host. This goes with degradation of bile acids, for example, production of short-chain fatty acids. You also have liposaccharides, endotoxins that are produced in the gut, absorbed by the host, and goes in the systemic circulation and can influence many organs in our body. That goes from the liver to the brain. And on the right, you can have an illustration of multiple diseases that have identified the microbiomes to be a major contributor to the disease. So you can see, for example, in the brain, you have autism spectrum disorder, stress in the lung with allergy, MASH, skin disease, type 2 diabetes, and inflammatory bowel disease. So that led us, to trying to understand what are the determinants of the microbiome. This is still an evolving field. They're still discovering many factors that influence the human microbiome. And one of them is the human genome itself. So this is just a summary of a study that had analyzed the microbiome composition and looked whether the human genome have any association with the gut microbiome. So you can see it's mostly in Caucasian population. There was a bit of South Asian in that study. And what this study have done is a meta-analysis. And what was found as a major result of that study with more than 20,000 individuals is that we identify 31 loci of the human genome contributing to the composition of the gut microbes. So this is what you can see on the left side with this Manhattan plot over here. And you can see on the x-axis is a chromosome. Each dot represents a given human genome single nucleotide polymorphism. So you can see it spread across the genome, but you have one particular SNPs or loci located in the second chromosome A. And this is indeed a gene that encodes for the lactase. And you can see on the right, the situation is quite complex. Because if you carry the wild type allele, you have a functional lactase production. And in that case, if you consume lactose, there's no symptoms of intolerance. And you can see that if you carry the recessive allele, you're not producing lactose. But if you carry specific bacteria that can produce lactose in vivo, in situ, you can still consume lactose and have no symptoms of intolerance. On the opposite, if you don't consume lactose and you carry the recessive allele, then that's when you start to experience lactose intolerance symptoms. So as I mentioned, there was 31 loci. Lactase was one of the major ones, but you can see that there is multiple association across the genome. And in many of those loci are associated with other diseases. One of them was located in FUT2. It's a gene that encodes for fucosylation. It can be fucosylation of the mucus, but also elements found in the breast milk. And those are key molecules that communicate with the host. And FUT2 is also a risk factor for inflammatory bowel disease. So for this rest of the talk, I'm going to use one disease as an example to study the microbiome. And in this case, I choose inflammatory bowel disease. So inflammatory bowel disease is a chronic disease that is predicted to affect almost 500,000 in the Canadian population, but it's also increasing worldwide, especially in Asia and South Asia. And the disease is believed to have different roots, including genetic, environments, immune response, and the microbiome. There is no cure for the disease, and we know that the microbiome is a major factor involved in the disease. And indeed, this is a recent paper published by Dr. Stewing from Hong Kong, showing in a very large cohort, multi-center, that in individuals that have ulcerative colitis or Crohn's disease, which are two forms of inflammatory bowel disease, they had lower alpha diversity compared to control. And on the right, when you look in a principal coordinate analysis, you can see where each dot represents the microbiome composition of a given individual. You can see that there is tremendous overlap in the three groups, meaning that IBD patients have overlap with a microbiome of healthy control. Although there is still significance, and this is indicated here in the PC axis, where there are some differences when you look at the microbiome in cases and control. The bottom part is just showing the major phylum, and you can see that there is a higher abundance of proteobacteria in patients with IBD compared to control, and there's also many other differences. However, when you do this type of analysis, looking at cases and control, it might be difficult to understand whether those differences in the microbiome are consequences of the disease or treatment of the disease, or those changes appear prior to the onset of the disease. So to answer that question, Dr. Kent Couture have developed the CCC GEM project, which aimed to identify the genetic, environments, and microbiome determinants of Crohn's disease, and have recruited about 5,000 healthy first generative of Crohn's patients that are prospectively followed since 2008. And this allows us to investigate what is different between the individual that goes on to develop disease versus the one who remained healthy. Multiple samples were collected, including blood, stool, and urine, as well as different questionnaires. And I'd like to highlight that whenever you're trying to better understand the microbiome, having a multi-omic approach to better understand the microbiome is really key to better understand what the microbiome is doing. And as I mentioned, we collected urine, which allow us to measure gut permeability. So this is an in vivo test where individuals are consuming a solution of lactulose, which is a big sugar that can cross the barrier if there's damage in the guts, and mannitol, which is more a reflection of epithelial function, surface area. So when we look at this data and trying to see whether the microbiome was associated with the gut permeability, we actually were able to find different microbial signatures that are present in individuals who have leaky guts. And indeed, we found taxa such as Aldecrotia or Cholidextrobacter that were increased in individuals who have an abnormal biofunction. And when you look at the microbiome function, we identify, I would say, classic pathways such as tryptophan and amino acids metabolism. And this was quite fascinating given the fact that we also showed that individuals who had a higher gut permeability were 3 times more likely to develop Crohn's. This is what is shown here in this Kaplan-Meier plots. The red are individuals that have had high barrier function, and blue individual who have a normal barrier function. So in terms of function, this is quite interesting because then what is the consequences of having an increased gut leakiness of the guts? Well, you may think that there is an increased translocation of microbial products, and this may trigger an immune response. And notably, Dr. Charleston in the University of Alabama have developed this array of antimicrobial antibodies that have many flagellated antigen, and found that this elevated antimicrobial antibody, specifically in the hinge region of the flagellum, is associated with complication of Crohn's disease. We apply this technology in the GEM cohorts, and we found that the individuals who later develop Crohn's disease have higher antimicrobial antibody response, especially in the hinge region and the flagellum, and mostly an IgG response. So what does that mean, and what can this lead to in terms of translational research? And so our collaborator in France, Dr. Benoît Chassin at Institut Pasteur in Paris, have developed a model of colitis. Those are wild-type mice that are colonized with salmonella, and those are the results of the animal receiving either water or different emulsifiers, so CMC or PAD. And those animals that receive those emulsifier, you can see there's an increased load of the flagellin, an increased load of LPS, and a reduction of the mucus layer, meaning that the bacteria gets much closer to the epithelial cells. But what was quite interesting in that study is if you immunize the mice with an anti-flagellin against salmonella, you can actually revert the phenotype. So you can see that the animals that are immunized for the anti-flagellin have lower flagellin in the stool, lower LPS, and you restore the distance between the bacteria and the epithelial cells. So perhaps that can lead to novel findings and perhaps vaccination. There's still a long work to do there, but it's opening new possibility. So what I did not mention yet is whether the microbiome itself contributes to the risk of developing inflammatory bowel disease. And indeed, when we look at the GEM cohorts, in this case, 3,000 individual, we apply machine learning algorithm called survival random forest that was developed in a training set. So we take 2/3 of the cohorts, and this subset of the cohort is used to train the model, optimize the model, and then we apply this model in the validation sets of 1,000 individual and 30 individual who go on to develop Crohn's disease. And what you can see on the right are the results of the microbiome risk score. And you can see that individuals who have a high microbiome risk score in red were twice more likely to develop Crohn's compared to the individual that have a low microbiome risk score. So with machine learning algorithm, you can identify which taxa contributes to the predictive performance, and we identify taxa such as Rhamnococcus torques that was increased in the individual who later developed Crohn's, but we also notice a reduction of Faecalibacterium in the same individual that later developed Crohn's. So to go a little bit beyond and understand the microbiome function, we also look at stool metabolites. And indeed, on this heat map, on the x-axis, you have the taxa that contributes to the microbiome risk score, the top 20. The microbiome risk score itself on the very right of the heat map. And on the y-axis, you have the metabolites found in the stool that are associated with disease. And this approach allows us to identify the most potential interesting bacteria. Notably, you can see Lactospira and Faecalibacterium have almost an opposite pattern compared to the microbiome risk score, which suggests that in terms of function, those two taxa are probably more important to reduce risk of disease. On the opposites, you have this taxa from Osteospiraceae, UCG02, that have very similar patterns to the microbiome risk score, which mean that potentially this bacteria is much more relevant functionally to explain the risk of Crohn's. So that was fantastic, but one of the drawbacks on looking at stool metabolomic data is you don't know if those metabolites are they there because they're not absorbed by the host, or do you saturate the system to a point where they can no longer be absorbed by the host? So we complemented this analysis by looking at serum metabolomic data, and we were able to confirm that this specific bacteria was correlated with many serum metabolomic marker of Crohn's. This includes peak result sulfates, phenylacetylglutamines, and others. So what is also key when you look at microbiome is to go a little bit beyond association. And indeed, association study are not causal study. And the way we address this question is using animal model. So we took advantage of the GEM cohorts once again, where we took 12 discordant siblings. So those are healthy individuals that are related, two siblings from the same family. They're both healthy. One developed the disease, one do not. And we took the stool of this individual, we transplanted this into germ-free recipient mice. And what you can see is the mice that are colonized with the stools coming from the individual who later developed Crohn's have higher histological damage, higher lipocalin 2, which is a marker of inflammation in mice, really supporting the fact that microbiome can transfer the inflammatory phenotype to germ-free animal. In terms of function, we confirmed that there was an increase of different fatty acids and sphingolipids that correlates well with inflammatory phenotype in the recipient mice. Those goes from weight loss, lipocalin, gene expressions, and different histological marker. But we also find in green, indicated here, some metabolite that's correlated with a protective role, notably different antioxidants or rhamnose degradation pathway. So I didn't cover all the topic today, but the microbiome, at least in inflammatory bowel disease, is central. There's multiple determinants of the microbiome, her host genome, her metabolome, diet is another aspect of this, permeability function, are all determinants of Crohn's, which in turn is also associated with disease. And what I'd like to mention is that the field of microbiome is moving towards shotgun sequencing. And what I mean here is most studies looking at microbiome are looking at this. So the top of the iceberg, which is 16S RNA sequencing, or IGS if you're interested in fungi, which is sequencing a very small fragments of the bacterial full DNA 16S, which is 1500 base pair. And we miss a lot of information here. And in comparison, when you look at deep shotgun sequencing, you have much more complete overview of the function of the microbiome. You also have much better resolution identifying what are the strains present in a given sample. You can also look at fungi and DNA viruses as well, but you can also interrogate your microbiome much more deeply, looking at directly the function. You can estimate growth rates of specific bacteria. Is there any antibiotic resistance genes, virulence gene, mucin degradation? You can estimate all of that with shotgun sequencing. And this is particularly relevant given the fact that there is more and more clinical trial that aims to modify the microbiome. And this is from 2022. The field there's even more study going on now. And you can see that a lot of the studies trying to aim to modulate the microbiome are from Europe and North America with multiple diseases. And on the bottom part, you can see that you have, in general, a good response for different diseases, including inflammatory bowel disease, C. diff infection, and cancer. So with all of that, it's become critical to develop new tools to modify the microbiome and perhaps alter disease course or maintain remission or prevent disease. And this is not an easy task to do. There's different approach to do that. You can either use a dietary approach of food supplements. There's also some treatments, and more experimental, like engineered bacteria or bacteriophage. And of course, you also have other techniques such as fecal transplants, with the goal is to basically reinitiate or reestablish a new microbiome in a given individual. Personally, I'm working more on the dietary aspects of how diet can be used as a tool to modulate the microbiome. And it's indeed because in previous work, it was shown that individuals who were eating a Mediterranean-like pattern were less likely to have high calprotectin, and about 50% of that anti-inflammatory effect was mediated by the microbiome. In a follow-up study, we also look at dietary fiber. Those are different fiber subtypes, and we found that individuals who were eating more inulin and beta-glucan were less likely to develop Crohn's disease. So to go a bit further, we also look at the microbiome, and of those individuals who are consuming different amounts of those dietary fiber. So those are the two associated with reduced risk of disease. And what was quite interesting is those fiber were actually reducing taxa that we found to be promoting or associated with increased risk of disease. And on the opposites, they were increasing the abundance of bacteria that we found to be associated with a reduced risk of Crohn's disease. So with all of that, I'd like to thank many people from the Crotour lab, notably Ken Crotour, the PI of the GEM project, my collaborators in Holy, as well as my collaborator for the statistician team, notably Osvaldo, the Verdu lab, Mark Silverberg, Jeremiah Faith, the founding and you for your attention, and thank you again for the invitation.
Thank you, William, for such a great talk and serve as an amazing introductions for us to jump into the next speaker. I would like to remind the participant that you will be able to ask questions in your Q&A tab in the bottom of the bar. So enter your questions and we will have the Q&A session after the second presentation. The second presentation is pre-recorded. It's presented by Dr. Premysl Bercik. The topic is on gut microbiome as a key regulator of intestinal motility. Dr. Bercik is a professor of medicine in McMaster University in Hamilton. His interest is in study of microbiome and interaction of the gut-brain functions. So I am going to pass it back to the the ESNM to play the recording, and then we will open the forum for Q&A at the end. And Dr. Bercik will also join us for the question period as well, along with Dr. Turpin.
Good evening or good afternoon. First, I would like to thank the organizers for giving me the opportunity to speak about the gut microbiota and intestinal motility. As we all know, normal gut function is a result of intricate balance and coordination between several systems, including smooth muscle, immune system, nervous system, as well as epithelium, which determine the function of the gut, whether it's permeability, secretion and absorption, visceral sensitivity, or motility. And we know that the gut is exposed to myriads of antigens and neuroactive metabolites which are coming from the luminal contents, whether it's commensal bacteria, pathogens, or even dietary antigens. And we knew already for decades that gut microbiota is an important player in determining intestinal motility. And here I'm showing you the data from a study which was published in 2001, in which the authors examined the effect of bacterial colonization in rats. And we can see the occurrence of the migrating motor complex, MMC, how it happens in germ-free mice, and rats which were colonized with lactobacillus and bifidobacteria. And it's these colonized mice where the interval between the consecutive MMC complexes is shorter. When looking at the overall results, the authors demonstrated that the colonization with lactobacilli, bifidobacteria, and also Clostridia decreased the duration of this interval, while the colonization with E. coli extended the interval duration. So inspired by this study, we have examined the effect of bacterial colonization in notobiotic mouse models, and examining different sections of the gastrointestinal tract, as in most of the previous studies, the attention was. First, we examined effect of bacterial colonization on gastric emptying. We used a video fluoroscopy technique in which diluted barium is gavaged into the mice, and pictures are taken at the baseline at 4 minutes later. And as you can see, there was no difference between different groups. Mice with colonized with SPF, specific pathogen-free microbiota, had the same results as germ-free mice, or mice monocolonized with E. coli or Lactobacillus rhamnosus. However, there were major differences in intestinal transit, which we assessed by a different technique, by the bead study, where 5 small metallic beads are gavaged together with dilated barium, and short videos are taken at baseline at 3 hours later. On the left, you can see a representative picture of mice with SPF microbiota, and there is only one bead which is located in the distal small intestine, in contrast to the mice who is germ-free, where you can see all 5 metallic beads within the distal small intestine. And on the right, you see the overall results, where the germ-free mice had the slowest gastrointestinal transit, which was increased in mice with SPF microbiota or monocolonized with Lactobacillus rhamnosus, and the fastest transit was found in mice with E. coli. We then examined the mechanisms which could explain these results, and the first we examined the release of acetylcholine, which is a major neurotransmitter involved in motility. And we found that there were marked differences between individual groups, and these were mainly present in jejunum. To correlate the results of acetylcholine with other neurotransmitters and gene expression, we performed nanostring analysis in which expression of over 100 genes related to neuromuscular function and immune function were assessed. And to our surprise, we found many genes which are related to the immune function, but from the neural ones, it was only VIP, vasoactive intestinal peptide, which was differentially expressed in mice with SPF microbiota or those who were germ-free. We then performed immunohistochemistry staining for both VIP and acetylcholine esterase. And we found that there were major differences in VIP expression, but again, we haven't seen any differences in acetylcholine transferase. And again, these ones were mainly expressed in the jejunum, but not in the colon. We then examined whether VIP was differentially expressed in different groups of colonized mice, and indeed we found that the highest levels were found in mice monocolonized with E. coli, and this was lower in mice with Lactobacillus rhamnosus. And in a way, these results paralleled the overall levels of intestinal transit. So VIP is a neurotransmitter involved in motility. We know that VIP acts on receptors located on smooth muscle to mediate relaxation, but there were also multiple studies which demonstrated that VIP can serve as a co-transmitter at cholinergic synapses in the small intestinal myenteric plexus and activate jejunal cholinergic nerves. Several studies also showed that mice deficient in VIP had decreased jejunal motility, while VIP administration can enhance intestinal motility. So we have then measured the co-expression of VIP and acetylcholine, and indeed we found close co-localization between these two neurotransmitters in our mice. We have then performed additional experiments which show that VIP administration indeed accelerated intestinal transit. It increased muscle contraction, while VIP antagonist decreased muscle contraction. In the next series of experiments, we examined the immune mechanisms which could underlie the differences in intestinal transit. For these experiments, we used wild-type black 6 mice and mice which were deficient in MyD88 TRIF pathway, which is a major recognition pathway for gut microbiota. And while in germ-free mice, there was no difference between the wild type and MyD88 TRIF deficient mice, after colonization, there was a marked difference with increased in intestinal transit in wild type mice and no change observed in MyD88 TRIF knockout mice. This was also paralleled by the expression of jejunal VIP with marked increase in E. coli monocolonized mice in wild-type mice. To better understand which immune cells are involved in this bacterial recognition, we performed 2 experiments using either fingolimab, which is a sphingosine 1-phosphate receptor modulator, which non-specifically inhibits activation and migration of immune cells, and cosalen, which is CCR7 antagonist, which specifically blocks activation and migration of dendritic cells. We found that while cosalen had no effect, fingolimab decreased the expression of VIP at the myenteric plexus. So we ruled out dendritic cells, so which other cells could be involved? And our attention we addressed our attention to enteric glial cells, as these were previously shown to be to serve as intermediators between immune cells and the enteric nervous system. So in the next experiments, we measured the expression of S100 beta and GFAP, which are 2 markers of enteric glial cells, and we found that they correlated or co-expressed with VIP at the level of the myenteric plexus. To get better mechanistic understanding, we then performed experiments when mice were pre-treated with gliotoxin, which inactivates the enteric glial cells, and we have measured the intestinal transit. And the mice which were treated with gliotoxin has markedly reduced intestinal transit compared to the placebo-treated mice. And again, this correlated with the expression of VIP with decreased levels of in mice treated with gliotoxin. So what we think is happening is that gut microbiota is being recognized by the innate immune system, with MyD88 TRIF pathway playing the key role. We know that glial cells are involved, and they what is happening that the VIP neurons at the myenteric plexus gets differentially modulated and determine the release of acetylcholine from the cholinergic neurons, which then affects the overall intestinal transit. We know that this control of motility is dynamic. So we performed experiments where we colonized mice with permanent colonizer, which is wild type E. coli. And you can see that there is no difference between 2 and 4 weeks post-colonization, both in the intestinal transit and the VIP levels. However, if we use oxotrophic E. coli, this is a E. coli which cannot survive or cannot divide in the gastrointestinal tract, we see marked increase in intestinal transit and VIP levels at 2 weeks when the bacteria are present, but at 2 weeks later, when the mice became germ-free again, the intestinal transit and VIP levels return to the levels seen in germ-free mice. Conversely, if we treat SPF mice with a combination of antimicrobials, we can decrease the intestinal transit and also VIP expression in the myenteric plexus, showing that this is really a dynamic system which can react to the changes in the luminal environment. Does it have any relevance to the clinical care? Well, in collaboration with Roberto Di Giorgio and his colleagues, we examined the levels of jejunal VIP at the mesenteric plexus in patients with chronic intestinal pseudo-obstruction. And you know that these are patients with severe constipation. As control samples, we use patients who underwent resection for non-complicated intestinal tumor. And here I am showing you that the VIP levels at the mesenteric plexus is actually decreased in patients with SIPO compared to the patient controls. So overall, our data suggests that VIP may play a key role in the modulating gastrointestinal transit in response to bacterial changes, and it happens not only in the animal models but also in patients. So I started speaking about patients with SIPO, which is chronic intestinal pseudo-obstruction, and you may all know that it's a rare and heterogeneous syndrome characterized by recurrent symptoms of intestinal obstruction. There are radiological features of dilated small or large intestine with air fluid levels in the absence of any mechanical occlusive lesion. And these patients have alterations in neuromuscular or mesenchymal structure of the intestinal wall or its extrinsic neurocontrol, which chronically impair tonic and propulsive motor function in one or more segments of the gut. So we embarked on a study investigating the role of gut microbiota in patients with SIBO, and this was a collaborative study with our Italian colleagues led by Roberto Di Giorgio. And we pulled samples from two cohorts from Italy and Canada, and first we showed that microbial profiles clearly differ between patients with SIPO and healthy controls, which is shown on the left. And when you look at the right, also looking at the individual, at the abundance of individual strains and genus, there is a marked difference between patients with SIPO with expansion of Escherichia, Shigella, and Klebsiella. And I would like to point your attention to the bar at the bottom, which is labeled as SIPO1, and this is our index patients about whom I would be speaking later. And you can notice that this patient has his microbiota is mainly composed by E. coli and Klebsiella. So the differences in microbiota profiles are not only about the composition, but also the metabolomic activity. So these are predicted metabolomic profiles inferred from 16S. And you can see there are multiple pathways which are different between healthy individuals and patients with SIPO. I would like to go back to our SIPO index patients, and when I first met this patient, he was 18 years old. He was transitioning from the pediatric gastrointestinal service, and he developed constipation at the age of 9 months, and his symptoms were gradually worsening. During these years, conventional treatments were not effective, and he had at one time also full thickness biopsies, which were suggestive of myopathy. He went through multiple surgeries, and finally he had full colectomy in 2011. And here you can see an X-ray from this patient, and it is actually after colectomy, so the dilated bowels actually are from the small intestine. So to investigate whether the gut microbiota has any role in the expression of this disease, we used his stool microbiota and colonized germ-free mice. And here I'm showing you a CT scan of mice colonized with his microbiota on the right, and compared to a mouse colonized with a microbiota from a healthy control. And what is highlighted in yellow, this is cecum. So you can see markedly distended cecum in mice with SIPO microbiota. And this also this was also true for the distension of the individual intestinal loops. The changes in bowel distensions were associated with changes in transit. So mice with SIPO microbiota had a much slower transit compared to the mice colonized with a healthy microbiota. We then studied whether the gut microbiota can induce changes in the host. And here I'm showing you the heat map of multiple neuroimmune genes from these mice. And as you can see, there are multiple genes related to the immune or neural system, which seem to be differentially expressed in SIPO microbiota colonized mice. And among them, there is nitric oxide, substance P, but also VIP, about which we were speaking previously. To understand whether fecal microbiota transplantation from healthy individuals could improve gastrointestinal transit, we first used this mouse model. So mice were either gavaged with microbiota from a healthy human individual, or they received their own microbiota. And while in the second case with the autologous FMT, there was no difference in transit, the mice which received the microbiota from healthy individuals markedly increased intestinal transit scores. And this was associated also with the decreased distension of the bowel, especially of the cecum. So encouraged by these results, we decided to perform FMT in our index patients. And I will first take you through the sequence of events. So this is the actually original picture of this patient prior to colectomy, with some multiple air fluid levels in the small intestine and colon. These are pictures when he was already under my care, and you can see up that after the colectomy, the small intestine was markedly distended. On the left at the bottom, you can see that his abdomen was also distended, and he was also malnourished. I mean, his nutritional status was very poor. At that time, he had already a gastric venting tube and a jejunal feeding tube, and we used the latter for FMT administration. We used the regime where initially the patient received FMT 3 times per week for 1 month. Then it was once time per week, and then for the last 3 months, it was only once per month. But you can see that at 20 weeks post-FMT, the bowel distension markedly decreased, and this was also seen when measuring the abdominal girth of this patient. We continued to follow this patient, and this is how he looked at 18 months post-FMT. And as you see, his nutritional status markedly improved. He remained basically asymptomatic for subsequent 8 years, and the patient is doing well, requiring minimal treatment in form of occasional prokinetics or mixture of probiotics when he feels unwell, but this is quite occasional. With regarding to his microbiota, here we can see how the microbial diversity was markedly decreased prior to FMT, and it increased within 1 week post-FMT, and it remained increased during the last 8 years. With respect to bacterial composition, I already told you previously that it was mainly linked to his microbiota was composed mainly by E. coli and Klebsiella. This was in marked difference with the healthy control. And his microbial composition markedly changed 1 week and 20 weeks post-FMT and remained within, I would say, normal or healthy range, even with 1.5 and 8 years post-FMT. So this is already my take-home message. So I showed you the microbial control of gastrointestinal motility is both region and bacteria specific, with major effects observed in the small intestine. It reacts to environmental changes and it's mediated by innate immunity neural system interactions. And we think that the enteric glial cells play an important role. We know that small intestinal VIP, by regulating cholinergic nerves in the myenteric plexus, plays a key role in this process. I also showed you that microbial profiles, both composition and function, differ between patients with SIPO and healthy controls. We demonstrated that SIPO microbiota can induce slow intestinal transit as well as bowel distension in notobiotic mice, and that FMT from healthy individuals can improve gut function not only in mice but also in our index patients. And in that case, this index patient remained asymptomatic for 8 years. So I think these are very exciting and encouraging data, and I think that the understanding of the specific mechanisms by which the microbiota affects the neuroimmune system is key to take advantage of its tremendous therapeutic potential. So at the end, I would like to acknowledge all people who were involved in the study, including Jade de Palma and Xiaopeng Bai, and all other co-authors and funding agencies. Thank you for your attention. I would be happy to answer any questions.
Well, thank you for the two presenter, and I would also like to thank the co-chair, Dr. Bercik, joining us from France, 1 a.m. in the morning. Now it's like 2. Why don't we start with the questions? There's 3 questions in the chat group. The first one is related to diet, so I'm going to put, although it didn't dedicate it to William, but I'm going to ask you. The question is milk good to consume or should we avoid?
Yeah, this is a quite difficult answer to say yes or no. I think it's going to be, it depends. I think milk have a lot of nutrients, but it's also digested by, in human, it's mostly bifidobacteria, and it's used, to for its central metabolism as a byproduct. You have acetate production, which is then used by other bacteria such as Faecalibacterium, which leads to production of butyrate, which is in general a good thing. But you can see there's multiple reactions that are involved, so it's a bit challenging to really give an answer. Is it good or bad? I think it's going to be highly dependent on your microbiome, your genome, and I think what I showed today is a bit the chicken and egg question, because it seems like if you carry the bacteria that degrade lactose, then there's no problem regardless of your genetic makeup. If you don't carry the genes to degrade lactose, but you carry the bacteria, then it's fine. But then the question is, how do you deal with becoming, let's say, tolerant to lactose? And perhaps that's where you need to do a strategy where you have milk, but also the bacteria that can degrade the milk.
Thank you.
Thank you, William. We have a question for you regarding the efficacy or absence of efficacy of FMT in IBD. You showed us some evidence of the role of microbiota in the onset of IBD, but what about the treatment, the impact on treatment, and especially FMT?
Yeah, no, that's also a great question. I do think there's always a bit of a challenge regarding FMT and inflammatory bowel disease, because the disease is really heterogeneous. With it can be mild, severe, ileal disease, colonic disease, and so right now we label all of this in a single bucket. And perhaps what the field needs to do is to look at specific classification of the disease. There's also a challenge from study to study with different donors of the stool microbiome. So the source of the microbes can also differ across study, which make it very difficult to have a standard procedure that is used across different study and have something that can be validated many times. There's also the recipient's microbiome as well, because some individual can be resistant to colonization, and sometimes what's needed is either antibiotic treatment prior to FMT, with can also influence have negative impact, and it can be very complex to tease out what is coming purely on the FMT or what is coming from the treatment around FMT. I think what's needed as well is long-term randomized clinical trial to really have definitive proof of the benefits of FMT in IBD, and it can be quite challenging. And I think if I remember, the last part of your question was about the prevention potential. I think it is very complex as well, because in terms of ethics and logistics, and how do you standardize the microbiome over time is also quite challenging.
Thank you. I don't know if we could contact Permec. Luis? We can't hear you. You're muted.
No, I tried it. I haven't been able to I tried to contact him, but he hasn't responded. So I just looked at the last question. It's also only 3 minutes to 8 o'clock. What may I suggest is that I can I can copy and paste these questions, and I can then send it to Dr. Bercik. My question to Alisa Tieg is that if we can, if you don't mind to share your email, or do we have the email of the person who asked the question? Because I can get the response from Chamek and then forward that to, so that they can contact an email. So then maybe that's the best way to get the answer for the questions.
Make maybe just a comment. The data that Permec showed on this presentation are intriguing. But I think that there is a confounding factor, which is that in patients with severe motility disorders, you may have bacterial overgrowth. So it is again a story of chicken and egg, and it's difficult. Well, the case he showed is very impressive, but I don't know if it is reproducible in other patients. And showing that microbiota plays a role is difficult, because in this patient there is a bacterial overgrowth, and I think it is a very important confounding factor. We have other questions arrived, I think, for 1 or 2 minutes. Yes, it is the email. Okay.
Perfect. So I think the next question is from Dr. Sino. She's asking that colonic versus small bowel pseudo obstructions, although that's I think related to Chamek as well. It's a jejunal and VIP with the isolated colonic pseudo obstruction. Is the driver jejunal microbiome or same for the small bowel involvement, or different mechanism with microbiome? I think these are as what's Frank said is an intriguing question. I don't think we have the clear answer to that, but the data that show was quite interesting that even in the simple patient that if I understand the data that he showed for the pseudo obstruction data, the evidence that showed that also the transit was in the small intestine. So it's a jejunal biopsy in Roberto's study that with Chamek.
Okay. So to conclude, thank you very much, William. We will thank Permek. I don't know why he couldn't join us because he was traveling, I think. So that's why he sent us a pre-recorded video. So thank you very much for attending this webinar. Thank you to William. Thank you, Luis, for co-chairing this. He's there.
Chamek
He's
logged
traveling.
in. So maybe you can answer those two questions for us. That
I'm sorry for joining late, but I couldn't get a signal. I'm here in Alberta Badlands, you know, so it's very unstable, the internet.
Yeah. So, well, thank you for joining. We're just about to conclude. Since you joined, we still have some participants, and let me make sure that the first questions was from Alyssa Tate. She's asking about the mechanistic evidence in terms of the microbiome intestinal transit. She has two questions. Is there any evidence using human cell line derived from neuronal tissue? And then he specifically asked the 2023 CMGH study that showed the effect of microbiome intestinal transit. Is it likely that could be similar impact on segmental motility, specifically related to the E. coli for the segmental dysmotility, and that related to the abdominal cramp in patient with irritable bowel syndrome?
So I am not sure about any studies on neuronal tissues, and I think it's a complex interplay. I mean, the study we published in 2023 suggests that there is really interaction through the immune system. So the glials, enteroglial cells are probably the intermediaries between the gut microbiota and mediating the effects on the myenteric plexus. With respect to the segmental effects, I mean, we haven't seen in contrast to the previously published studies, much effect on the colonic motility, but in our experiments, the major effect occurred in the small intestine, I would say distal jejunum, proximal ileum. We don't have many studies in humans on this, and is there any evidence that this could be used as a probiotic? Maybe. I think that we have to understand really very well how the individual bacteria signal towards the immune and the neural systems. So I think that the 2023 study was very basic, and I think we have to push further to understand what the complex communities are doing.
Yeah, thank you, Chamek. And Maria Sino in Toronto also paying close attention to your talk. She asked a very interesting question regarding to the isolated colonic pseudo obstruction. Based on your what it showed us about the importance of jejunum and the VIP in the study, is the driver of the colonic motility from the jejunal microbiome, and it's the mechanism because so the question is related to the specific colonic pseudo obstruction. Is it the driver from the jejunal micro dysbiosis?
So with respect to the chronic intestinal pseudo obstruction, we don't know the mechanisms yet. We know that when transferring the microbiota from that patient to mice, we can reproduce the phenotype. There are multiple effects on the enteric nervous system. We were trying to identify the metabolites which would be responsible, and so far we have failed. So I think that we need further studies. This is one isolated patient, but I think the results are impressive, and I think that it warrants further studies. And probably the next one should be an RCT and see whether the FMT could be really beneficial in these patients, and probably in a proportion of these patients it may really help. And I mean, you all know, I mean, we don't have many other tools to, I mean, what we can offer to these patients.
Great.
Thank you so much for joining, even if it was a bit late. But I think we have to conclude and to thank again the two colleagues who presented very, very interesting and very recent data. And thank you so much for that. And thank you for attending this webinar. And I wish you a nice day. And now I will go to bed.
Good night.
Good night.
Goodbye.
Bye-bye.
Bye-bye.